Chapter 9 Biomolecules, Short Notes and Summary from Class 11th, Subject Biology. Introduction. Fundamental observation. Although both living tissues and non-living matter like Earth's crust contain the same elements, the relative abundance differs. Living tissues have a notably high proportion of carbon, hydrogen, and oxygen. Biomolecules defined. Biomolecules comprise both small organic compounds, micromolecules, and large macromolecules that form the building blocks of cells and perform critical metabolic functions. Focus of the chapter. The chapter introduces diverse chemical compounds found in life, from simple metabolites to complex polymers such as proteins, nucleic acids, and polysaccharides, and explains how their structures relate to their functions. 9.1 How to analyze chemical composition. Elemental analysis. A sample, plant tissue, animal tissue, or microbial paste is analyzed to list the elements present. Although the same elements are found in non-living matter, living tissues have higher relative amounts of carbon and hydrogen. Separation into fractions. Acid soluble pool obtained by grinding the tissue in trichloroacetic acid contains thousands of small, molecular weight 18 to 800 Da organic compounds. Acid insoluble fraction obtained by straining the slurry contains macromolecular compounds like proteins, nucleic acids, and polysaccharides, and also membrane fragments that include lipids. Additional analysis. Drying a tissue to remove water and burning it to yield ash helps determine the inorganic composition, example salts and minerals. 9.2 Primary and secondary metabolites. Primary metabolites, found in all animal tissues, include essential compounds such as amino acids, sugars, and lipids, directly involved in normal growth, development, and metabolic processes. Secondary metabolites, common in plants, fungi, and microbes. Examples: alkaloids, example morphine, codeine, flavonoids, rubber, essential oils, antibiotics, pigments, scents, and spices. Not directly involved in basic metabolism but have roles in ecological interactions and human applications. Example: pharmaceuticals, flavorings. 9.3 Biomacromolecules. Definition and characteristics. Molecules with high molecular weights, generally 10,000 Dalton and above, predominantly found in the acid insoluble fraction. Major classes: proteins, polymers of amino acids, nucleic acids, polymers of nucleotides, DNA and RNA, polysaccharides, long chains of monosaccharide units. Special note on lipids. Although many lipids have lower molecular weights, less than 800 Dalton, they occur as part of cell membranes organized in vesicles, which makes them migrate with the macromolecular fraction during analysis. 9.4 Proteins. Basic structure. Proteins are polymers called polypeptides made by linking together 20 different amino acids via peptide bonds. Key points. They are heteropolymers because their chains contain a variable sequence of different amino acids. The sequence of amino acids, the primary structure, determines the protein's higher level structures and functions. Functions: serve as enzymes, hormones, antibodies, transport molecules, structural components, etc. Dietary proteins are essential to supply amino acids that the human body cannot synthesize. 9.5 Polysaccharides. Definition: long chains of sugar molecules, monosaccharides. Types and examples: homopolymers, cellulose, made entirely of glucose, provides structural support, example in plant cell walls. Energy storing polymers, starch in plants and glycogen in animals. Branched or helical polymers used for energy storage. Other examples: inulin, a polymer of fructose found in certain plants. Structural features: presence of reducing and non-reducing ends. Starch helical structure, help it bind iodine I2, producing a blue color. 9.6 Nucleic acids composition, polymers made of repeating units called nucleotides, nucleotide components, nitrogenous base, purines, adenine and guanine, pyrimidines, cytosine, thymine in DNA and uracil in RNA, sugar, ribose in RNA or 2-deoxyribose in DNA. Phosphate group links consecutive nucleotides together to form the backbone. Functions: DNA carries genetic instructions, RNA involved in protein synthesis and gene regulation. 9.7 Structure of proteins. Levels of structural organization. First, primary structure, the linear sequence of amino acids N-terminal to C-terminal. Second, secondary structure, localized folding patterns such as alpha helix, right-handed coil stabilized by hydrogen bonds, beta pleated sheet, extended strands connected via hydrogen bonds. Third, tertiary structure, the three-dimensional folding of a single polypeptide chain stabilized by hydrophobic interactions, hydrogen bonds, disulfide bridges, etc. Fourth, quaternary structure, the assembly of multiple polypeptide subunits, example hemoglobin composed of alpha and beta subunits. Importance: each level of structure is crucial for a protein's biological function. 9.8 Enzymes overview. Enzymes are biological catalysts that speed up chemical reactions by lowering the activation energy. Most enzyme molecules are proteins, though some nucleic acids, ribozymes, can also have catalytic activity. They are crucial for metabolic pathways, enabling rapid conversion of substrates to products without being consumed. 9.8.1 Chemical reactions. Definition: chemical reactions involve breaking and forming bonds. Key concepts: reaction rate, the speed at which products form per unit time; activation energy, the energy difference between reactants and the transition state; catalysis, catalyst including enzymes accelerate reactions by lowering the activation energy barrier. 9.8.2 How do enzymes bring about such high rates of chemical conversions? Mechanism: active site binding, substrates bind to a specific region, active site of the enzyme; formation of the ES complex, the enzyme substrate ES complex stabilizes the transition state, reducing activation energy. Example: carbonic anhydrase speeds up the conversion of CO2, carbon dioxide, and H2O, water, into carbonic acid by millions of times. 9.8.3 Nature of enzyme action. Enzymatic cycle. First, substrate binding. The substrate binds to the enzyme's active site. Second, induced fit. Binding induces a conformational change in the enzyme, ensuring tighter binding. Third, catalysis. Chemical bonds within the substrate are broken and reformed to form products. Fourth, product release. The product is released and the enzyme returns to its original state, ready for another cycle. Key point: the high specificity of enzymes is a direct function of the active site structure. 9.8.4 Factors affecting enzyme activity. Temperature. Every enzyme has an optimum temperature. Deviations, especially high temperatures, can denature the enzyme. pH. Enzymes operate best within a narrow pH range. Changes can alter the ionization of key residues in the active site. Substrate concentration. Increased concentration speeds up reaction rates until all enzyme active sites are saturated. Vmax is reached. Inhibitors. Chemicals that bind to the enzyme, example competitive inhibitors, can block substrate binding and reduce activity. 9.8.5 Classification and nomenclature of enzymes. Enzyme classes. Enzymes are broadly classified into 6 categories based on the reaction they catalyze. First, oxidoreductases catalyze redox reactions. Second, transferases transfer functional groups between molecules. Third, hydrolases catalyze hydrolysis reactions, break bonds by adding water. Fourth, lyases remove groups from molecules to form double bonds. Fifth, isomerases catalyze the rearrangement of atoms within molecules. Sixth, ligases join two molecules together using energy from ATP. ATP full form is adenosine triphosphate. Nomenclature: each enzyme is assigned a unique EC, enzyme commission, number that reflects its class and reaction type. 9.8.6 Cofactors. Role in enzyme function. Many enzymes require additional non-protein components, cofactors, to function. Types of cofactors: prosthetic groups, organic molecules tightly bound to the enzyme, example heme in peroxidase. Coenzymes, organic molecules that transiently bind to the enzyme during the reaction, often derived from vitamins, example NAT positive. Metal ions, ions such as zinc 2 positive or magnesium 2 positive that stabilize the enzyme substrate complex or participate directly in catalysis. Key insight: removal or absence of these cofactors results in the loss of enzyme activity, underscoring their essential role. Finally, summary: chemical composition and analysis. Living tissue contains similar elements to non-living matter but is enriched in organic elements CHO. C is carbon, H is hydrogen, O is oxygen. Chemical analysis separates compounds into acid soluble micromolecules and acid insoluble macromolecules fractions. Categories of biomolecules: small molecules, metabolites include a diverse range of organic compounds involved in metabolism. Macromolecules, proteins, nucleic acids, and polysaccharides are polymers with high molecular weights and specialized functions. Lipids, while low in molecular weight, form part of cell membrane structures. Proteins and their structure. Proteins are made of amino acids and possess hierarchical structural levels, primary to quaternary, that determine their unique functions: enzymatic, structural, transport, etc. Nucleic acids and polysaccharides. Nucleic acids, DNA and RNA, encode genetic information, whereas polysaccharides serve as structural elements and energy stores. Enzymes and catalysis. Enzymes accelerate biochemical reactions by lowering activation energy. Their activity and specificity depend on the precise structure of their active sites, and they are influenced by environmental factors, temperature, pH, substrates, inhibitors. Classification of enzymes into 6 main groups and the indispensable role of cofactors highlight the diversity and regulation of catalysis in living systems. So friends, we have completed short notes and summary of chapter 9 Biomolecules. We will do question answers of this chapter in a separate video. If you like the video, kindly like it and share it with your friends. Also subscribe to my channel. We will meet in the next video. Thank you for watching.